presentation, please go ahead and send in your questions, upvote the questions that you want to see answered if other folks have already submitted a question that you want to see answered. We are going to respond via chat during the presentation and also verbally after the presentation. I want to say a big thanks to Jack, who works on Survive the Sound Communications and arranged this amazing tech for us today, and he's helping out with technical support on this webinar as well. So if you have issues, he's the Q&A to go to. I'm Iris. I'm a senior project manager at Long Live the Kings, and I serve as back-end data support for Survive the Sound. So I'll be telling you a little bit about the science behind the game. But first, I want to hand the mic to Jacques, our executive director, to say a few words of welcome.
Thanks, Iris. Thanks so much, everybody, for participating. This is really great. This is a kind of a new thing for us to be running an information meeting with 100 participants online live. So bear with us as we work through this. I think you're going to enjoy Iris's presentation, and hopefully we have a robust question and answer period afterwards. Our goal today is to discuss, review the race this year, discuss some key findings from our work on steelhead, and to answer some of your questions about Long Live the Kings, about our work and our partners' work to recover steelhead, and how Survive the Sound came to be. Iris is going to do a great job talking about the background and the direct connection to our technical field work, but I just wanted to tell you a little story about how this got started. We were in a board meeting discussing the results of some of our work on recovering steelhead in Hood Canal that involved the technology that's behind the game. And one of our board members who is involved in a gaming company said, hey, that's so awesome. You should figure out a way to bring that to the public and turn it into a game. So what you participated in and experienced this year and over the last 4 years, this is the 4th year we've done it, was the fruition of that idea. And we had no idea, honestly, how to do that until we started to partner with Vulcan. And they had some technical resources that allowed us to take the technical information about the fish and turn it into a public engagement game, which we think is pretty great. So I'm going to be available with Iris after her presentation to answer questions about our organization, about our work, and about, to the best of our ability, about salmon and steelhead in general. So enjoy, and thank you so much for participating. Take it away, Iris. Uh-oh, Iris, I don't hear you.
Hey, there we go. I had the mute so I wouldn't disrupt you, and then I forgot to turn myself back on. Thank you all for participating in Survive the Sound 2020 and joining us today. Thanks, Jacques, for that intro. Now I want to take you on a special tour behind the scenes and let you in on some of the real life experiences of our favorite fishy racers. So like Jacques said, this is our 4th year running the Survive the Sound game, and it has been by far our most successful yet, with over 20,000 participants and over 2,000 teams. You all raised over $40,000 to support wild salmon and steelhead recovery, and that is frankly just amazing. Again, thank you so much. As you all learned while playing, this game is based on real life data from these 48 incredibly cute little steelhead. So this game is possible because of our ongoing research and monitoring projects with our partners at NOAA Marine Fisheries Station. Every year, Megan Moore and I go out to the river. We catch wild steelhead and we surgically implant a tiny acoustic tag into their bellies. So here's a picture of our tagging setup on the banks of the Nisqually River. We've got this table, we've got a tent because it was really sunny that day. And our little steelhead friend here is nestled into a cradle that we built especially for tagging. There's a little hose that goes into his mouth, and that pumps anesthetized water over his gills. Once the tag is in place, Megan stitches him up. The incision is really small, so it only takes 2 tiny little stitches, and we use the same sterilized surgical materials that you would get stitched up with if you, had a fall and hurt yourself. And then this little guy, after that surgery, goes straight into the recovery tank. The surgery takes about 2 minutes when Megan and I are working at full capacity, and these fish recover really fast. They're usually up and swimming within seconds. We keep them in the recovery tank overnight just to make sure that they are fully recovered and ready to go back into the river. And then we release them back to the river to continue their migration out to the ocean. As they migrate, they have these tiny tags in their bellies. They emit an acoustic signal. We call it a ping. And we have a network of receivers that are shown here deployed throughout Puget Sound that are listening for and recording those pings. Each of these tags has a unique ping, so we can track each fish individually as it moves past those receivers. So now you know how we get the data that creates the Survive the Sound experience. Let's take a closer look at what happened to the fish in this year's game. Maybe. There we go. We started with 48 determined little racers from 3 watersheds in Puget Sound, the Skokomish River that drains into Hood Canal, the Nisqually River in South Puget Sound, and the Green Duwamish that's right in our very own backyard in Seattle. By the end of the outmigration, you all might have noticed, of those 48 little fish, only 7 survived to make it to the ocean. We had 3 survivors from the Nisqually, 2 survivors from the Skokomish, and 2 from the Duwamish. As you and your teams probably discovered, and as this map of death locations makes clear, there are unique challenges that fish from each of these watersheds face. So today we're going to go behind the scenes with 3 of these little fish, Ocean Magic from the Skokomish, April from the Nisqually, and Grape from the Duwamish, and they're going to show us in detail some of those challenges. So let's start with Skokomish, with Ocean Magic's journey. If you had a Skokomish fish on your team, this pattern is probably going to look really familiar to you. Okay, so here he goes. He's coming out through the river. He's doing really well, gets to the estuary, moves up Hood Canal, right up the center of the canal, passing by our Lily-Walk field station. Shout out to Rick and Joy and the work that they've done to recover steelhead in Hood Canal for over 2 decades now. Coming up past Dabob Bay, still doing really good. Go Ocean Magic. And he's approaching now the Hood Canal Bridge. He gets to the bridge and he kind of starts doing these back and forth behaviors. Like, doesn't really know where to go, maybe got hung up at that migration barrier, still trying to get past. Oh, he got past. Nope, he came back around, still doing these back and forth behaviors. Almost, and nope. Oh, he didn't make it. So this pattern probably looks familiar to you if you're a Skokomish fish, because almost every single Skokomish fish, even the survivors, ran into issues at the Hood Canal Bridge and navigating that barrier. So what's going on? Well, we did 2 years of intensive fieldwork through the Hood Canal Bridge assessment to figure it out. We actually have a video up on our YouTube page that goes into more detail, so check that out later if you're interested. Essentially, the Hood Canal Bridge is a floating bridge, the longest saltwater floating bridge in the world, and it's supported by these giant concrete pontoons that are filled with air. They span about 83% of the width of Hood Canal and extend about 15 feet down into the water column. And that doesn't sound very deep from a human perspective, but our research shows that juvenile steelhead like to stay in the top 3 feet of the water column when they outmigrate. So as these little fish are approaching the bridge, from their perspective, it kind of just looks like a big concrete wall. Overall, about half of our steelhead that make it to the bridge, that's this little segment here of the migration, die there. And that's really, really high mortality to see in just one little spot. So we put a lot of effort into figuring out why that's happening. Do you remember in that video we just watched how Ocean Magic had these back and forth movements along the bridge? It turns out that is actually a really typical pattern for non-survivors. So this is an animation of several tags that we know died at the bridge. Some were in the game, some were not. And you'll see that they're all doing these back and forth movements. This isn't a normal steelhead behavior at all. We're pretty confident that this is actually a predator behavior. So it's a predator that's eaten a steelhead. The tag is transmitting from within the predator's belly as it does this sort of patrolling behavior up and down the bridge infrastructure, probably looking for more food. We have some clues about who these predators might be. We were able to deploy in our Skokomish and Big Beef Creek fish some tags that were depth sensitive and temperature sensitive. So here's an example of a depth sensitive tag that's showing a really typical steelhead behavior, very surface oriented, right at the top of the water column. And then all of a sudden, it starts making these deep dives down to 40 or more meters. And that's definitely not a juvenile steelhead pattern. So we know that these predators are diving predators. Now here's an example of a temperature sensitive tag. Steelhead are cold-blooded. If you want to sound fancy, you would say the word poikilothermic. And that basically just means that when the tag is in the steelhead, it should be the same temperature as the ambient water surrounding it. But this tag here starts with that ambient water temperature, and then it heats up to about 95 degrees Fahrenheit. And that indicates that this fish got eaten, likely by a marine mammal. Many of these tags showed predator signals like this, and all of the tags that showed predator signals later became stationary, meaning that we know the fish that were tagged with these tags died. These data and other research that our partners at Washington Department of Fish and Wildlife and the Port Gamble Squalum Tribe have done at the bridge and in surrounding areas suggest that harbor seals are playing a big role in contributing to steelhead mortality at the bridge. Ocean Magic's journey is an example of that fate. That tag made it to the bridge, was delayed there, got eaten, started showing these back and forth predator movements, and then finally it got deposited right near Sisters Rock, which is a seal haul out just south of the bridge. The conclusion is pretty obvious there. Okay, so now let's see what happened to April, migrating out of the Nisqually River in South Puget Sound. And here she goes, heading down the river, looking good so far. She's getting towards the estuary. She's crossing under the I-5 corridor, and she starts doing these kind of back and forth movements, going in and out of the estuary and up and down the river, and then eventually dies in South Puget Sound right next to the estuary. So for Nisqually fish, I mentioned that overall survival to the ocean was slightly higher. 3 of those 16 fish survived versus 2 from the other 2 rivers. Even though their overall survival to the ocean was slightly higher in this year's game, several of the Nisqually fish showed this kind of pattern in the estuary. So let's take a little bit of a closer look at what's going on there. So this is a tag. This is not April's tag, but this is a tag that is behaving very like April did. That's displaying this upstream, downstream, back and forth movement, essentially moving along with the tide. The color coding on this tide graph, the tidal height graph, shows these locations in the estuary as well. So red is further out into saltwater, green is further upriver. And you can see here that as the tide comes in and the saltwater intrusion extends further up the Nisqually River, the tag is moving up the river too. And as the tide goes out, the tag moves downriver with it. This is not a steelhead pattern, so already we can start to get a little suspicious. Well, this particular tag started doing these kinds of movements in early May, and then something really interesting happened. In late May, another tag started doing this. And it's not coming up on the slide, but I promise it will in a minute. There we go. Another tag started doing this, and it was detected at exactly the same places, exactly the same time. And then in early June, a third tag started moving alongside those 2. So these tags, as they appear on the screen, we're pretty sure are all together in the belly of a predator that's doing these upstream, downstream movements with the tide. Now, these 3 tags were all pinging continuously from the moment that they were put inside the fish. But as part of another study that we did in the same location, we also had deployed tags that we programmed to be silent for the first several days of outmigration and then turn on at a specified date. And here in early June, this is a tag that was programmed to be silent until June 2nd. And it turns out when that tag turned on and started pinging, it was already in the belly of the same predator and moving right alongside those other 3 tags. Now, this didn't go on for long because then all 4 tags were deposited right over here in South Puget Sound, all together in one little lump. I bet that you can guess where this story is going. If you guessed seal predation is a challenge for steelhead in the Nisqually River, then your guess is spot on. You are correct. There are several places that seals like to haul out in South Puget Sound, even in the estuary itself. In this picture, it's kind of hard to tell that there are even seals there from this distance. They kind of look like little grains of rice all clustered together. So let's move a little closer. And if you moved closer still, closer than you would ever want to get to a seal in the wild, so please don't go this close. But this is what you would see at those haul outs. Seal abundance in Puget Sound has increased about 3-fold over the past several decades, in large part due to their success after the Marine Mammal Protection Act was written into law. But the story is a little more complicated than that. So some of our current research is suggesting that when other preferred prey, like forage fish, herring, and anchovies are plentiful, seals will fill up on those instead of steelhead. And steelhead marine survival rate through Puget Sound in years of higher forage fish abundance is actually much better than in years of low forage fish abundance. So supporting healthy forage fish populations could play a big part in helping steelhead survive to the ocean. I should also note that we are now starting to deploy temperature-sensitive tags in the Nisqually estuary as well. And while I don't have any of those preliminary data in these slides to show you today, all of the evidence is supporting this harbor seal marine mammal predation in the estuary story. It's even a little more complicated than that, though, because we've also started seeing more transient orcas coming into Puget Sound in recent years. These transient orcas, unlike our southern resident killer whales, which only eat salmon, these transient orcas eat seals and sometimes play with them, as you can see in this GIF, and they might impact the seal population by reducing their abundance. Well, that might be good for steelhead, but initially, we're also seeing some preliminary evidence that orca presence can cause seals to change their behavior. So in years where we saw higher transient presence in Puget Sound, we also saw seals sticking closer to the near shore and estuaries, probably to try to escape those transient orcas. So if seals are spending less time in the saltwater and more time near shore in the estuary, that might actually increase predation risk on steelhead. So it's a little bit of a complicated scenario, and we are currently working with our partners to better understand this story and understand what can be done about pinniped impacts in Puget Sound. So finally, we come home to the Duwamish and our little tiny steelhead racer, Grape. And here he goes. Now he's going to start by going the wrong way up the river. He actually goes upriver a little bit, takes his time coming down the river, and dies when he gets to the estuary. So there are a couple things that might be going on here. We're going to talk about both of them. The first thing, and this is something I think that is very well known among the salmon community, and probably most of you since you love salmon, the estuary habitat is really critical to juvenile salmon. It provides shelter from predators. It provides food for the fish to grow nice and strong. And there is evidence suggesting that the bigger a fish can grow in that early life stage, the better its chances of surviving in the ocean. And it also provides, shelter from predators, food, and it also provides this transition to saltwater. So as a fish comes out the river, down the river into the estuary, there's a mix of freshwater and saltwater coming from the river to the ocean. The fish needs time to prepare for that transition. Physiologically, the estuary habitat is really important. Now here are two estuary habitats in Puget Sound. The top one is the Skagit River, which has a mostly intact estuary. It's a really healthy system. The bottom is the estuary that our friend Grape came out of, the Duwamish estuary. Duwamish estuary has lost a lot of critical habitat over the past several decades, and it is fairly impacted as an estuary. You can see that visually in these photos. If you were that tiny little salmon, you know which estuary you would prefer. Long Live the Kings actually has a new project working at this site where the star is, along with Vigor and the University of Washington's Wetland Ecosystem team, to restore 2.64 acres of estuary habitat and monitor how that affects the vegetation, the insects, and the fish that use this area. So maybe next year, Grape will do a little bit better. The second issue that might have happened for some of your Duwamish racers, that these fish were secretly part of an experiment. We didn't highlight this in the game, so this is very behind the scenes. We found evidence through research done through our Salish Sea Marine Survival Project of potential parasite impacts in a few watersheds throughout Puget Sound, and the Duwamish is one of them. So these fish were a treatment control experiment where we raised fish. We had them in two separate groups in the laboratory. One group was not infected by parasites. The other group we infected with parasites. And then we put tags in both groups and saw which fish did better. I'm going to tell you a little bit about the parasite. Its name is Nanophytis salmincula, but we typically call it Nanophytis or Nano for short, just for ease of saying. So here's the life cycle of the nanoparasite. It starts in an embryonated egg. It goes to a snail host. The snail host eats the eggs. The parasite eventually emerges from the snail host as a free-swimming cercaria, which then penetrates the skin of the fish and actually gets into the gills, the heart, and liver tissue, or sorry, kidney tissue. Later on, it gets into a marine mammal or a mammalian host, and the parasite continues its life cycle. But we really are caring right now about the cercaria penetrating the skin of the fish and affecting its internal organs. So here's a couple pictures of fish that have been exposed to this Nanophytis parasite. If you cut it open, you can see that it will encysst itself, typically in the posterior kidney, creating these little tiny sort of cyst structures that potentially impact the fish's swimming ability, and at very high concentrations can actually cause mortality. So as I said, we infected half the fish, we left the other half alone, and we put them all in the river. And we learned through this that overall, the survival was fairly similar to the ocean between the infected and uninfected fish, but actually in river survival was much lower for infected fish. These fish spent a lot longer in the river. They had a harder time transitioning out to the estuary, and we think that that's because this parasite at higher infections can influence that saltwater transition. The fish is already compromised, and the additional stress of physiologically adapting to saltwater is just one straw too many for that poor little fish. And that may be what happened to grape. So all this work cannot happen without your support. Thank you so much for playing Survive the Sound. It's been just under half an hour, so I've left plenty of time for questions. I am not following questions yet, so I will go and look at the Q&A, and I'm sure Jacques has some queued up for me. But again, thank you so much for playing Survive the Sound this year, donating to Long Live the Kings, and advocating for salmon recovery. It is truly essential.
Oh boy, Iris, we have lots of questions.
That was so much information.
So I'm going to run through them.
I didn't know if I could get through all that.
No, great job. I'm going to run through them, and people keep continuing to ask questions as they come up, or if you have questions about our answers to the questions, please provide those too. So let's not waste any time here. Most of these are for you, Iris, but I'm going to take a couple of them. So the first question is, given that this is from Deborah, and it's given that the steelhead are out in the ocean for several years, how do we track the fish when they return, or can we track the fish to see how they do when they come back?
Yeah, that's a great question. So I'm going to give you the short answer and the long answer. The short answer is that our partners at Washington Department of Fish and Wildlife and our tribal partners, monitor spawning grounds and have a really good information on how many fish come back. The longer answer, with these specific fish, that we won't know about these specific tagged fish. So the tags that we use in these fish are VEMCO tags. They're V7s usually, variations on the V7 tag. That means they're just about the size of my thumbnail, and they have a little battery inside them. They can only ping for as long as that battery is active, and a battery at that size cannot last that long. So these tags will only ping for a couple months at maximum before they die. The technology is always improving, so it's possible that in the future we might be able to track returning adults as well that were tagged as juveniles. But with this particular type of tag and the technology capability right now, by the time these fish come back as adults, those tags will not be pinging. We won't be able to track them specifically.
Okay, great. So the next question is—
if that doesn't answer it, please add a follow-up.
Thanks. The next question is from Serena, and she's a 6-year-old. And her question is, why so much mortality before reaching the ocean?
Hi, Serena. It's amazing that you're playing the game as a 6-year-old, and I hope you learned a lot about the game. The mortality in Puget Sound is largely a result of all of those factors that we talked about today. When fish are compromised, meaning that they're sick coming into the salt water. So maybe they have a parasite, maybe they have been exposed to contaminants, maybe they have a disease. If fish are sick, it's really hard for them to swim well. It's also really hard for them to escape predators. So a sick fish is more likely to get eaten than a healthy one. And then the sheer number of predators and the decrease in alternate prey sources. So if a seal is out there and he wants to eat a fish, he's going to choose a herring if there's a herring there because he loves to eat herring. But if there aren't enough herring, then he'll eat whatever he can find. And often that's salmon because they're about the same size and they're also silvery, just like herring are. Does that help?
Well, I hope so. So then we have a, thanks Iris. We have another question, and this is from two different people, from Anonymous and from George. And the question has to do with what is the mortality rate, or how many died at the Hood Canal Bridge? And I think you answered that in your talk, but why don't you go ahead and answer that again?
Yeah, so it varies a little bit each year, but we've seen pretty consistently that about half the fish that get to the bridge will die there. So I think I had it up on one of the Hood Canal Bridge slides. For 2 of the years that we measured, I think we were at just under 50%, and the other was just over 50%. Those are the only 2 years where we had a VPS array at the bridge, so we could really triangulate where exactly those fish were dying. But in other years where we have had receivers, but not that fancy array, we can still look at survival, and those years, it's ranged from about 50 to 65% death at near the bridge.
Okay, thanks. So here's a question from another anonymous.
Sorry.
Oh, go ahead.
Jacques, can I do one quick follow-up?
Yeah,
I just
please.
glanced at the question and I saw that Eric Carlson asked, is the sample size large enough to be credible? And I would say yes, the 48 fish that you are seeing in the game are not all of the fish that we tagged. We have tagged over 500 fish in each system. And in Nisqually, we've tagged many, many more than that. We've been tagging there since 2006. I'm actually going out tomorrow to tag fish in the Nisqually. So the program continues. We are tagging 176 fish this year. 76 of those are going to be temperature sensitive tags, and 100 will be our regular Vemco V7s. So I would say yes, statistically, our sample sizes are good. The stories that I'm telling you today are anecdotes, but they are supported by statistically sound science.
So here's a couple of related questions, Iris. One is from Anonymous, and it says, how many Duwamish fish survived? And then there's another related question from Margaret that says, how do the 3 basins compare in terms of survival?
Yeah, good questions. Okay, so the first one, in the game, 2 of the 16 Duwamish fish survived. In real life, our control fish, so I have the river mouth to Strait of Juan de Fuca survival rates. That's only the marine survival rate, not the in-river mortality. So our control group that weren't infected with the parasite had 14% survival to the Pacific Ocean from the river mouth. Our treatment group, I wish I could tell you the in-river survival off the top of my head, but I can't. I don't have it in front of me. However, it was higher than the control group, and then their marine survival, the river mouth to Juan de Fuca survival, was better at 23%. So the 2 groups kind of evened out by the time they got to the ocean. The mortality just happened earlier on for the treatment group. And that second question, sorry, comparing among the basins. It's weird to say this, but survival is actually pretty comparable among basins. And that's been true for several of the populations that weren't included in this game that we've done previous tagging work in. Typically what you'll find is that the longer a fish has to travel through Puget Sound, the lower the survival chances are. So Duwamish fish, we would expect to have a slightly higher survival rate than Nisqually fish, for example. And in 2014, we actually did a transplant experiment where we took some fish and released them from the Nisqually and some fish and released them from the Duwamish. And the origin of those fish was a mix from both rivers, so we didn't have a river confounding effect. And we found exactly that the fish that had to travel farther through Puget Sound and be exposed to potentially higher predation risk in South Puget Sound did very poorly, much worse than fish from, for example, the Skagit, and a little bit worse than fish from the Duwamish. Fish from Hood Canal have to travel about the same distance as fish from the Nisqually, but they actually survive really well in Hood Canal until they get to the bridge. And that makes us think that if we were able to help them pass the bridge, that we might see quite a good improvement in survival there. Does that answer?
Thanks. Thanks for the questions, everybody. So here's another one for you, Iris. And I think you answered this in your presentation, but let's do it again. This is from Elizabeth.
Say it again.
And it said, and she asked, why do fish go back and forth instead of straight out?
Yeah, that back and forth behavior is almost certainly a predator behavior rather than a true steelhead behavior. The typical steelhead behavior is straight out to the ocean, right at the top of the water column. And even when they're in the ocean, they do tend to be a little bit more surface oriented than other adult salmon species. Usually if you're seeing that back and forth in the estuary or that back and forth at the bridge, we assume that's a predator behavior. And our temperature sensitive tags and our depth sensitive tags have really confirmed that for us, that when we see those atypical behaviors, those are predators, not fish.
Okay, hope that answers Elizabeth's question. Here's another one for you, Iris, and this should be easy. This is from Pat, and they ask, is data from last year?
Yes, so these tags, they ping, and the receivers, they pick up those pings, but it is not real-time data. We have to actually go out and pick the receivers up and download those data and process them before we can get a sense of what the fish are doing. So the data for this game is always either last year or the last couple of years, depending on when and where we were able to tag. And this particular year's game, we didn't get to tag last year in Skokomish, so those data are a mix of 2018 and 2017. For Nisqually, it's all 2019 data, and for the Duwamish, it's all 2019 data. So mostly last year, yeah.
Okay, so here's a question from Jace, and I'll take this one, Iris, and give you a break. So Jace asks, would it be better to have orca whales in Puget Sound to eat the predators, seals and sea lions? So I guess if you're a seal or a sea lion, no, not so much. But it's definitely one of the things that we're thinking about. So in the Marine Survival Project, we have identified that at least harbor seals and perhaps other predators are eating juvenile Chinook, coho, and steelhead. And so we've been thinking about what are some of the solutions that we could employ to address that. Now, in the early and middle part of the 20th century, there was a bounty on seals and sea lions, and you could kill them and bring in the snout and get paid, and that was pretty effective at controlling the entire population. But that was also a pretty dramatic and drastic approach, and given the Marine Mammal Protection Act and some of the sensitivities of the human population here, that's not the first solution to come to mind. So one of the first solutions that does come to mind is, I think Iris mentioned this, the change in the transient or big killer whale populations in Puget Sound and the Strait of Georgia. Those populations have been increasing along with, or perhaps lagging a bit behind, the increases in the seal and sea lion and harbor porpoise populations. So one of the things that we really would like to get a handle on is what would it take for the transient killer whales to bring the pinniped and the porpoise populations back into some kind of balance with salmon production and salmon survival. So that is definitely something that we were looking at. We need more data. It doesn't really look like at the current abundances that that's happening. If anything, the seal population has plateaued or is increasing slightly. I will also say that there isn't any evidence that sea lions are eating juvenile salmon or steelhead. That sea lions tend to go after larger prey. They have very large gape and teeth, and perhaps a juvenile steelhead could be food for a sea lion, but we don't see a lot of evidence for that. And the data that we have comparing stellar sea lions, California sea lions, killer whales, and harbor seals for their ingestion of Chinook indicates that sea lions are much less than killer whales and seals, with seals focusing on the little fish and killer whales focusing on the big fish. Some other things that we're looking at in order to control predation by sea lions, I think Iris mentioned the importance of forage fish. Increasing forage fish populations is a double win. It provides an alternative food supply for predators on steelhead and salmon. It also directly feeds Chinook and perhaps some coho as they rear in Puget Sound. So that's a plus. So looking at things like restoring forage fish populations is a possible solution in addition to looking at whales. Also looking at our hatchery practices. If we're dumping fish out of hatcheries in the millions all at once, that could be ringing a dinner bell or sending a signal to predator populations, whether they're birds or other fish or marine mammals, that it's dinner time. Whereas in natural populations, fish tend to migrate out of rivers more spread over time and not in huge slug when we just open the gate and let the hatchery fish out. So that may have a benefit to the hatchery fish in terms of their survival, to look at different release strategies. It may also help them as they experience different food availability in the environment. If our environment's changing, changing hatchery practices is important. And then lastly, looking at barriers where we put haul out structures at the mouth of rivers where it's easy for predators to get access to out-migrating juvenile fish. And also, as we've noted, the Hood Canal Bridge seems to be a real hot spot for mortality. So that's a long-winded question, but that was a really long-winded answer, but that was a really good question. And I think a last resort would be looking at specific control of predators by humans where some of those other solutions aren't working. So here's a question.
I think there are a few follow-ups in the,
Iris.
oh, sorry. I think there are a few follow-ups in the question about what we can do at the Hood Canal Bridge. And I want to highlight to you all that the Hood Canal Bridge impact assessment actually has two phases. The first phase that we're just finishing up now was the research phase, where we really tried to figure out what the problem was and get a handle on that. And those are the data that I talked a little bit about to you today. Now that we have a handle on the problem and we see how important it is to get those fish past the bridge without delaying their migration and to potentially deter the predators that are in that area, we're moving forward into phase 2. And phase 2 is exactly what you all are asking in the questions. Engineering solutions that help fish get past the bridge faster and keep predators away from the places where they are seen eating fish. So stay tuned, we're working on it. We have support through the state legislature, and we are hoping to start putting solutions in the water within the next several years.
So was that, Iris, was that an answer to this question? What engineering projects can help steelhead bypass the bridge?
Yes.
Yeah. I just want to say to folks, thank you so much for all of your questions. We're now up to 43 questions. I don't know if we're going to be able to get through all those, but we will.
We still have 20 minutes.
We will do the best we can. We'll go on a first come, first served basis to a point, and then we will default to upvoting. So if you really want a question answered and you look at and you scan through the questions, as Iris said, please vote for that and make sure that it gets answered. So Iris, Eric has a question here. Oh, you already answered this. Is the sample size large enough? What's the mortality rate? Okay, so here's a question
we also, for those of you who are scientifically minded and really want the details, I can send you our publications. Contact me later.
Okay,
so, it has all the statistics in it.
So here's a question that I'll take, and Eric asks, with Hood Canal mortality, why not focus recovery efforts elsewhere? So I'll just say that a lot of folks look at Hood Canal and the habitat that is there on both sides of the canal, both on Kitsap County and on the Olympic Peninsula side, where you have essentially a road and a little bit of development, but then you have federal forest lands and the national park. This is an area that should be a salmon stronghold and a place where we should be seeing very, very strong salmon and steelhead populations given the size of the basins there. And so there's a lot of interest in us in trying to recover those populations as a place compared to the east side of Puget Sound, where we have lots and lots of human development, agriculture, roads, buildings, pollution. Hood Canal is a relatively pristine area of Puget Sound. And so we are all working extremely hard to try to recover and protect those populations. So this is a sort of a regional decision, regional priority. The second issue is that steelhead are listed under the Endangered Species Act, and the Endangered Species Act does not allow us to pick and choose which basins where steelhead are currently occurring that we are going to recover those fish. So we are under some obligation to recover populations in each one of the, what we call major population groups. So there is a legal constraint. There's also sort of a common sense or a conservation mandate that says that we really ought to try to focus on recovering these. And given that we've identified what the problem is with the bridge, we think that, you know, if we can build societal interest in supporting the funding for that kind of work, that could be an important activity. So let's see, another question here. This one's for Iris. Have you gotten, this is also from Eric, have you gotten any negative feedback on anthropomorphizing the steelhead game?
None that I've heard.
And I'll take this one. And then a follow-up question was, how do kids react with their fish dying? And
oh, we've had some tears. We've had some tears.
Disgust,
it is
anger.
heartbreaking. I mean, I've cried when my fish has died. That is not actually true because I know the outcomes of the fish beforehand. But it is rough on all of us when we, you know, place our hopes in a fish and he doesn't make it.
I don't,
Megan and I
just to let
actually
know
in the
people know
field,
the game isn't rigged. My fish have died in the first day of the last 2 years.
That's
so.
because I keep all of the outcomes a secret. I'm very good at that. Megan and I will be in the field and we'll be like, oh yeah, this one looks really healthy. He's going to make it all the way. And then later we'll look at the tag data and be really disappointed. So it's heartbreaking for everyone of all ages.
Maybe the big ones make a better meal. So here's a question for Iris, and then I'm going to ask a follow-up question to it. So this is from Anonymous, and the question is, do you plan to infect fish next year? And my follow-up question is this harmful to other fish or the environment?
So we don't currently have plans for another treatment control nano study. This last year was actually the second year that we did that. And with the replication of the 2 years, we feel pretty confident in our results. That's not to say that there won't be nanophytus projects in the future. We have current work going on with our partners at the U.S. Geological Survey to understand how to treat nano in hatcheries and also identify hotspots in rivers to understand what mitigation actions could be taken there to reduce the parasite infestations and potentially impacts to the fish. And as to your follow-up, Jacques, this parasite cannot be transmitted fish to fish. Once the free-swimming Cercaria burrows through the fish's skin and insists, it stays in that fish in its cyst form until it's eaten by its final mammalian host. So if the fish can survive that burrowing in and infestation process and successfully transition to saltwater, he's good. He's not going to infect any other fish.
So this is related, maybe you just answer this. Here's a question, I can't remember who this is from, but what can be done about natural parasites in the Green Duwamish? And I would also add maybe the Nisqually River.
The Nisqually. Yeah, I didn't mention it during my talk, but nanophytus is definitely also a problem in the in the Nisqually River. And I should actually say that to give some context to the potential problems there, in the literature, before we started doing all of this work, the prior literature on nanophytus suggested that fish might experience adverse health effects with infection rates of 200 cercaria per kidney. In the Nisqually fish, for a few of them, we were seeing up to 8,000 cercaria per kidney, which is, you know, at that point, do you even have a kidney? It's just all cysts. Sorry, I got lost envisioning the cysts in a kidney. So mitigation actions that could be taken. As part of our work with USGS, we did do several potential treatment options focused on what you could do as a hatchery treatment, because we know that Stews Creek Hatchery on the Green Duwamish has had nanophthalitis problems in the past. And there are several different treatments that are effective at killing this parasite. Many of them are in use at hatcheries already, such as formalin treatments. Salt water will actually kill this parasite. So if the fish can get down the river without getting infected and get into salt water, she's safe there too. Ways that you could mitigate in river in an uncontrolled environment, unlike a hatchery. So a big part of this parasite's life cycle is this shedding stage, where the snail host sheds parasite, sheds the free-swimming cercaria into the water. And that shedding stage is, we think, based on our evidence, controlled mainly by temperature and light. So things like restoring foliage, natural foliage near a riverbed in a hotspot might contribute to keeping those cercaria loads down, preventing the fish from getting as badly infected.
So Iris, here's a question that has 3 upvotes, and it's from Michael. I think that's how you pronounce it, with 2 L's. And they are asking, this might be a better question for our project manager for the actual game, Lucas, but I'm going to ask you this, Iris. For fish that you are pretty confident about their cause of death, is there a way to include or see how they died in the interactive game map when they die?
I think we should talk to Lucas and the devs about maybe we could put a little pop-up or something by each fish. I take notes when I do the back-end data. I do take notes on what the probable cause of death is. And for some fish, you know, we don't know, but for some fish, we do feel pretty confident. For those fish, maybe a little pop-up message to let you know it's possible.
Okay. And here's a question from exec director issaquahfish.org. And this question is, any chance that fish can be placed in the water above the bridge?
Do you mean like barging around?
I'm going to guess
like
that's
taking fish from one
the
side
idea.
and putting them on the other?
Barged or trucked around.
I think that ties into another question that I saw. The question from Elizabeth: when engineers are designing bridges, do they take into consideration out-migrating salmon? And yeah, so this is also in our YouTube video, so you can definitely go check that out later if you're interested in getting in depth. But the Hood Canal Bridge was actually designed originally to accommodate out-migrating salmon. The problem is that in the late 1950s and early 1960s, what we understood about out-migrating salmon was that they always hugged the shoreline when they out-migrate. So the bridge is designed with these 2 open spans right along the shorelines to allow for salmon passage. We didn't find out until much later that steelhead didn't really do that steelhead don't hug the shoreline, that they go out into open water and hang out in the middle of Hood Canal. And so we didn't even know it was an issue until we realized that. So that's not a fault of bridge design or engineering. That's just we didn't know at the time. And exec director of Issaquah Fish, or whoever has that email address, your question about placing fish in the water above the bridge, we don't have any plans to physically pick the fish up from the south side of the bridge and put them on the north, but several of our engineering concepts for phase 2 of the Hood Canal Bridge Assessment are pretty much designed to do exactly that, to sort of guide the fish past the infrastructure around the open span. And back to the center of Hood Canal, or sorry, of Admiralty Inlet on the north side of the bridge.
So Tess has a question here that's been upvoted 3 times, and it has a lot of parts to it, so I'll try to parse it out and simplify it. I don't know if we can answer all these, but she says she's interested in learning about her fish in particular. And the question she has is her fish a girl or a boy? Her fish is named Willie. And was the fish infected? Did the fish get to spawn? She notes that the fish was one of the 7 to survive and wondered if it could tell a little bit more about why her fish survived and others didn't. I think you maybe have answered that last question, but maybe we'll have to get back to them about that. You probably don't have the information about
Yeah,
the
I don't have Willie right at the top of my mind, but I can definitely look at the data and let you know. Your first question though, Tess, about whether your fish is a girl or a boy, that one I can't answer. So for 2 reasons. One reason, when salmon are that small in the smoltification stage, so the stage at which they're transitioning from freshwater to saltwater, their gonads, so their sex organs, are not fully developed. So even if you opened them up and looked inside, you wouldn't be able to tell the difference until they're a little bit older. The second part to that answer is, if we cut it open and looked inside to find out whether it was a girl or a boy, the fish would not survive that. So we would have to kill the fish to find out. Fish on the outside, or at least salmon, you really can't tell whether they're a girl or a boy from the outside until they hit their adult stage and return to the rivers in their spawning colors. And then you get some sexual dimorphism. And what that means is that the males will look different from the females at that point.
Okay. Here's a question from Anonymous that has 2 upvotes, and the question is, how long does it take a steelhead to transition from fresh to salt water?
Our steelhead is pretty fast. It's a lot faster than perhaps for some other salmon species that outmigrate smaller at earlier sizes, or earlier at smaller sizes. Pretty much all of our steelhead are basically ready to go once they get to salt water. It might take, a few hours for them to feel comfortable in that transition. But part of that is because they do stay really close to the surface as they outmigrate. So they are probably taking advantage of the freshwater lens. Freshwater has a different density than saltwater. You can think of it like freshwater kind of floats on top of saltwater as they outmigrate through Puget Sound, which is essentially one really, really big estuary if you think about it the right way, because there is freshwater and saltwater input into all of Puget Sound from all of our rivers around the sound. They take advantage of that freshwater lens and stay really close to the surface, maybe dipping down a little bit here and there as they get closer to the ocean. So you could think about their outmigration as this long transition, if you'd like. And if you're thinking about it in that way, it's about 2 weeks to get from river mouth to the ocean.
So here's a question that got 2 upvotes from M.A. Fleming. And this one, I think you partly answered, but you might want to link back to this question from your discussion about the temperature tags and what that tells us. So the question is, can you tell yet when a smolt gets eaten by something other than a seal, porpoise or bird, for example?
We can't. I wish we could. We are pretty confident in what we call seal behaviors, in part because of the temperature and depth sensitive tags that I've talked to you about, in part because we've consulted with lots of folks who are experts on pinniped behavior, and in part because in 2014 and 2016, we actually tagged seals as well. So we had seals that we could literally actually see what they were doing and where they were going. And those seals also detected steelhead tags. It was very cool. We don't have good data yet to let us know what it looks like when a bird or a porpoise eats a steelhead. The majority of predator behavior that we have identified in these tags, we would attribute to seals, but there are certainly some tags that die that haven't shown seal behaviors before that death. And that's likely another predator or a seal predator that we just didn't see the behavior in. We don't have any way of picking that out at this point.
So here's a question from Matt that has 2 upvotes, and I'll start this, Iris, and you can add value as you see fit. In all the years that the game has played, have the Nisqually fish survived better? ? I'm wondering, since I assume that the Nisqually estuary is in better shape than the estuaries in the other 2 rivers monitored. So answering the last part of that first, the Nisqually has probably the largest single estuary restoration project in Puget Sound, so it is in good shape, although the Skokomish River estuary also has some pretty major habitat restoration projects recently that may, as a group, add up almost to the same level of habitat or providing the same levels of habitat as the Nisqually restoration. So those two rivers are more comparable. Although the Nisqually fish don't have to go by the bridge, they seem to have to go by a very robust seal population in South Puget Sound in the Nisqually estuary. The Duwamish River estuary used to be a 5,300 acre wetland, and now you're lucky if there's 100 acres of wetlands in that river estuary. It's a major port, a major industrial area, sports complexes. It's certainly not what it used to be. I think that in terms of the survival, I'll let Iris answer that. But I think she already did kind of address that question. Now, I will also say this: steelhead tend to swim very quickly once they make the commitment to go into the estuary and into marine environment. They don't linger there. Estuaries are the most important for rearing juvenile Chinook. And so areas where we have good estuary habitat seem to increase the survival of out-migrating Chinook, particularly Chinook that migrate out of the river early as smaller fry as opposed to larger smolts. And so that estuarine habitat is a bigger, more critical factor for our Chinook populations, and particularly our wild Chinook populations than for steelhead. And I'll let Iris add any value to that she wants to.
Yeah, what Jacques is saying is entirely correct. That estuary habitat is really critical, especially for sub-yearling Chinook, especially for the smaller ones that are coming out that need that time to grow and those resources to grow and transition to saltwater. I wanted to talk about the importance of estuaries because I think it's a super, super important thing for folks to understand. But it is true that our steelhead are not spending a whole lot, or they wouldn't be spending a whole lot of time in the estuary if they weren't in the bellies of seals. The Nisqually estuary is incredible. It's, from all we can tell, really healthy in many ways. And the fish that are coming out of that system are huge. They're the biggest steelhead that we tag, sometimes even up to 300 millimeters, which is crazy. They're just monsters. They look like they ought to survive really well. In some years, in more recent years, when we've had those higher forage fish populations, yes, Nisqually survival has been better than the other populations. And you saw that in this year's game where we had 3 fish surviving from Nisqually and only 2 from the other rivers. And that was accurate to real world survival rates. In other years, earlier in our data set where the forage fish were not abundant in South Puget Sound, survival for Nisqually fish has historically been really, really low. In the late 2000s, we were down to something like 6 or 7% survival for those fish, which is incredibly low.
So I think we have time for about one more question, and this is a good one. It's been climbing up the charts in terms of upvotes, and this is it. I'll let Iris answer this. How do you come up with the fish's names? And this is from an anonymous attendee.
Well, we had a lot of fun with that. So the first year, Jacques mentioned that this Survive the Sound game, the project itself, was an idea from one of our board members, Gaylord Kellogg, and he's a great guy, genius for inspiring this, really. We all sat down as a staff the first year, and this is before we knew how successful this game could be. We were just, trying to brainstorm and spitball and come up with something that we could do to gauge interest and see whether folks liked it. So we sat down and we said, okay, well, if we were steelhead, what kind of funny fish puns would we be? And we came up with a huge spreadsheet of fish puns. And then we started working with a talented illustrator called Jocelyn Lee. She's on our website, and we have linked to her design work in other projects as well. And she took the fish puns that we came up with and illustrated them, and it moved on from there. In subsequent years, we also had y'all's help. So we ran a contest where one of our prior year's winners designed a fish. Ocean Magic was actually that fish that got designed by one of y'all. We had a contest where a school kid got to design a fish. I think we ended up with Lunchbox and Gryffindor out of that. And we have some sponsors who have been very generous in their support of Survive the Sound and enable the program to continue, and they get to have a say in their fish. So Grape, for example, was sponsored by Chinook Wines. So Chinook Wines came up with that name. Mostly it's just a lot of fun.
So I just want to thank everybody for participating in Survive the Sound, for taking your time to come on and watch our info presentation today. And I want to thank Iris for her great presentation and her great answers to everybody's questions. This is a lot of fun. We're going to do it again next year. If you have more questions for staff, you can contact us at info@lltk.org. And we thank you very much. We really appreciate your support and your engagement. And we're all in this together to restore wild salmon and steelhead and support sustainable fishing in the Pacific Northwest.